I think we were going to cover the Facebook Meta announcement that their AI research team had generated the physical structure of 617 million proteins from these metagenomic data sets. And so remember, AlphaFold made this big announcement that they had highly accurate predictions of protein structure, the three-dimensional shape of proteins. And remember, proteins are kind of the machines of biology that do everything from catalysis to enzymes where they break stuff down. And they're like, they have all this structure that allows them to do specific physical things. And proteins are coded in DNA. Every 3 letters of DNA codes for an amino acid. A string of amino acids makes a protein. And so, we have about 1 million species where we've sequenced the entire genome of those species. Only about 3,000 animals, by the way, including humans. 500,000 from bacterial species, and then a bunch of viruses and other stuff. But call it about 1 million species that we've sequenced. And so, earlier this year, Google's AlphaFold project published the 3D structure of 200 million proteins that they had derived from the whole genome databases that existed, where we've gone through and figured out what's the full DNA sequence of all these different species. Now, when you look at the DNA in the environment around us, you were just to take the DNA out, it turns out that we have seen very little of that DNA. The vast majority of DNA that you would find in a teaspoon of soil, for example, we've never classified. It's not part of a species that we've actually built the whole genome around. We may not even know what species that DNA is from. And so when you take a teaspoon of soil, you'll get about 100 billion microorganisms in that soil from about 1 million unique species. But you don't see those species because the way DNA sequencing or shotgun sequencing works is DNA is chopped up into little 250 base pair lengths, little 250 strands. And those 250 letters are read at a time. And then statistically, bioinformatics puts together all of that little DNA segments and tries to create long strands of DNA to figure out what the genes are or what the whole genome is. And so shotgun sequencing gives us kind of a snapshot of the DNA. But until we've done the hard work of figuring out the whole genome, we don't know what species that DNA comes from. So when you take a sample of soil or you take a sample of human poop and you sequence it, or even a teaspoon of ocean water, and you just sequence the DNA in it, you get all of these little segments of DNA that we've never seen before. And you can string them together statistically because you get lots and lots of copies of them, and you can figure out the overlap, and then you can create these genes. And a gene is a segment of DNA that codes for a protein. And those genes make up the metagenome, or the combination of all the genes that we find in a piece of the environment. And that metagenome comes from millions of species that we've never seen before. So what they did at Meta is they took all of those genes that we pull out of the soil or we pull out of the ocean, and they picked a bunch of random samples. And they then predicted the physical structure of the proteins from just those genes without knowing what organism they came from. And this gives us a whole new universe of proteins that we've never classified before or never seen before. Now, I will just kind of speak a little bit critically about it. Number one, they didn't do what AlphaFold did. What AlphaFold did is they took 3D structures from typically X-ray crystallography. Then they took the DNA code and they built machine learned models to figure out the 3D structure from the DNA code. What these guys did at Meta is they took the 3D structures and the code, and they basically did a fill in the blank. They found all the metagenome data out of these samples, and a lot of it was missing. And they filled in the missing blanks using kind of common protein structure that existed out there in the wild that we already knew from the AlphaFold data. And so they kind of did a fill in the blank. And as a result, it allowed them to very quickly build these 3D models versus doing the hard and rigorous work that AlphaFold had to do. So they claim that it was 60 times faster, but it's actually an entirely different technique. And the second thing is that they represent that only about a third of it is high quality, meaning only about a third of the proteins that they've created structure for are really useful or that could be kind of applied in terms of this is the real representation. Now, why is this interesting and important? Proteins can form the basis of new medicines. So, we can find proteins in the soil, genomes in the soil and proteins in the soil that can kill certain fungal pathogens, that can kill bacteria. And those can be turned into fungicides. They can be turned into antibiotics. We can find proteins that bind to specific things. We can find proteins that fix nitrogen from the atmosphere, and those proteins can be turned into new types of fertilizer. So, searching through this universe of proteins that exists in the metagenome will allow us to find new molecules to do new and interesting things with in the applied engineering world. And I will say, like, this is...
What would the output of these be is what everybody's going to be thinking.
Antibiotics, fertilizers. I mean, the idea of the metagenome is rather than start with the species and then take the genes out of it, just go get the genes. The genes are already there. They're in the ocean, they're in the soil, and there's millions of genes, hundreds of millions, billions of genes that we've never seen before. Therefore, there's billions of proteins. Now, we could randomly create proteins, but the number of proteins that could exist is more than the number of atoms in the universe. Because remember, there's 20 amino acids, so 20 to the 200th power, or 20 to the 300th power, or 20 to the 1000th power, meaning how many different combinations of amino acids can you make? That's more than there are atoms in the universe. So the best place to start is what evolution has already given us, all the proteins that exist in the environment. So let's go find those proteins in the environment, and then let's figure out, what do we think they can be used for? Can they be used in industrial applications, in medicine? Can they be used in... We could be
doing them in material science, you're saying, correct?
Material science. And so, a lot of drug discovery mines proteins. It tries to find proteins and figure out what can these proteins be used for. And now we have all these new data sets of proteins that are being generated from these metagenomes. And so it's amazing. I mean, look at the world around you, look everywhere, up and down, on the walls, on the ground, below you. There are billions of species of organisms that we've never classified before that are making billions of unique proteins that we've never classified before. And any one of them could unlock an amazing commercial opportunity for industry, and for medicine and for human health and so on. So that's what's really exciting about this ability to kind of mine the metagenome.
Silly question for you, or maybe not silly. We have gotten the precursors to DNA from meteorites, if I understand correctly. What do they call them? Nucleobases?
Nucleic acids?
Yeah.
Nucleic acids?
Thymine, et cetera, like things that exist in DNA that are precursors. We've never had DNA from space, obviously, but we could at some point start to find DNA out there in space, and this could have an even crazier impact on what we built here. Is that the next card to turn over after we human genome what's here?
No, I wouldn't say so. I think, look, if there's DNA that's coming to us from meteorites, call it a couple hundred genes. You could pick up a piece of soil and find over 1 billion genes in that teaspoon of soil, right? So we have far more to mine here on Earth. And the low cost of DNA sequencing and shotgun sequencing, coupled with bioinformatics, where we take all that. So just to give you guys a sense, when you take a teaspoon of soil and you get the DNA out and you read the DNA out of it, sequence the DNA, that's potentially tens of gigabytes of data. And then you could do that millions of times over. And then you statistically can find genes and then statistically estimate what they physically look like, what those proteins look like. And then you can start to build models around which ones do we want to try and use for drug applications? Which ones do we want to... So there's so much work to do just in terms of what we have here on Earth. And the tools are getting so cheap and so available. There are labs that are all over the world starting to kind of spring up to do this work. It's super exciting.
Chamath, any thoughts?
It's kind of, just to put two ideas together. I've said before, like the two big investable themes that I'm orienting my organization around is this. One is that the marginal cost of energy goes to zero. And the second is that the marginal cost of compute goes to zero. And the second one is really about shifting compute to more parallelism on GPUs and ASICs and FPGAs. But that's why all of this stuff is possible. The fact that, Meta can do this and Google can do AlphaFold is largely because the cost of all of this stuff is, trivial for these kinds of companies. So it's really exciting. It's going to move science, in my opinion, out of this in vivo, in vitro experimentation model into silica. And so those who can actually build learning machines will solve some of the most important biological problems. So I'm a real believer in this stuff. I think it's super exciting.
When do you think this stuff actually hits Friedberg, our life? That's always when people talk about these discoveries.
A lot of people don't realize it, but so many molecules that are used in agriculture, like fungicides to kill fungus in the fields, those are derived from this sort of work. A lot of antibiotics, a lot of medicines are already derived from mining genomes, finding new proteins, and seeing what those proteins can do. Because these proteins didn't evolve in the environment randomly. They evolved to do something. And in many cases, we can take that thing that they do and then harness it into a product. And that's what's so exciting. And this affects everything: material science, agriculture, human health. It's a food. It's really profound.
Awesome. All right, everybody. There you have it. That's another all-in podcast in the can.
So, by the way, so many of Freeberg stands were afraid, Jay Cal, that you and I were going to make some joke, and we didn't.
No, we didn't.
And so, for all these stands, I hope you guys can exhale,
take a deep breath, man.
have some, eggless mayo, and enjoy your weekend.
Enjoy your week.